The allied seed and variety improvement industries are critically important aspects of vegetable production systems in California. The state is home to a number of both public and private sector efforts that have done much to enable truly state-of-the-art vegetable production to thrive in California now for many decades. The people who work in a range of ways to improve vegetable seeds and crop varieties are part of a very dynamic, specialized, and quite complicated enterprise, and are a very important segment of sustainable vegetable production.
Well, my name is Kent Bradford, and I'm a professor in the Department of Plant Sciences at the University of California at Davis. I'm a seed biologist. I'm also the director of the Seed Biotechnology Center here at UC Davis for the past 18 years. Most vegetables are annuals. There are a few perennials, but for the most part, vegetables need to be planted every year, so they have to start from seeds. So the seed development, breeding, seed production, distribution, and so on is extremely important for the vegetable industry. All the crops that are possible biologically to make hybrids, for example, so that is hybrid seeds where you have two distinct parents that are crossed then to make seeds that are sold to the farmers. Makes this process even more complicated because now you're breeding both parents, you're making a cross every year in the field. Often that needs to be done by hand, believe it or not. So for peppers, tomatoes, things like this, a lot of that goes on with a lot of hand labor, actually removing the pollen or emasculating the female plants, putting pollen on, tagging those flowers, collecting those fruits, and so on. Not always. We have many that can be made in the field. Brassicas, for example, all of the broccolis, cabbages, and so on. Those are all hybrids, but have other ways of manipulating the fertility of those plants to make hybrids. But the point is that the process of making seeds for vegetables is very complicated, very involved. Perhaps the most interesting one is triploid seedless watermelon seeds. Those plants that produce seedless watermelons do come from seeds, so it's very tricky to make seeds that then produce seedless fruits. The process of developing new varieties and then making seed, particularly in vegetables, is highly technical and quite a complicated process. The breeding starts back, for example, even in wild species or related species that can be bred. For example, a major target of all breeding in vegetables is disease resistance. Often this is the most limiting factor in production. Spinach right now, for example, is having terrible problems with downy mildews, and the main way to approach those is through new varieties. So breeders start back with wild germplasm if necessary. Make crosses and back crosses to their cultivated lines until they can incorporate traits that they need. At the same time, they have to be also aiming for production traits. They have to be growable, harvestable, high yielding, and they have to have consumer traits in mind. What do consumers really want? And that varies greatly. Most vegetable seed breeding companies operate globally. That is, they operate in 100 to 120 countries around the world. And watermelons are a good example. In some parts of the world, people like really big watermelons with lots of seeds, and or else they're not considered to be good. Here, for example, little small watermelons, seedless, that fit in your refrigerator have really disrupted the market and have taken over. So consumer needs and wants are very important. Flavors becoming more and more important to consumers. And in the meantime, always focusing on disease resistance, insect resistance, production traits that growers have to have. So then what they actually do is a lot of crosses. That is, they make sexual crosses between the plants, transfer whole genomes essentially, and then sort them out. So it's basically, all breeding is basically a process of creating variation and then narrowing it back down. So once they have that and have a variety, if it's an open pollinated variety, we have, for example, lettuce, other crops where it's very difficult to do anything but just let the plant self. So we have beans, lettuce, and so on that are largely self-pollinated varieties. It's a matter then of growing the seed and producing that seed in a very careful way. Making sure that it's been rogued, meaning off types are removed and so on, and to keep the seed pure. For hybrids, it's a whole different thing, where then during the breeding and development, they're breeding both parent lines, male lines and female lines, which then when combined give additional uniformity and additional vigor and productivity. That can be interesting and tricky in certain cases to make that all work out. So the breeders have to know the breeding pools, so the two parent lines and so on. In that case, that's generally because it's possible to do hybrids in some ways. So for example, by insects, that makes that easy, but those same mechanisms probably mean that in the parent lines you have to do hand pollination. So in some cases it's easy to maintain the parent lines, sometimes it's easy to make the hybrid, but there's always one step that's probably by hand or more difficult to do. And so all of those things have to be done. Some crops, in addition, you think of onions. Onions are a hybrid crop. It's actually one of the first vegetable hybrids ever developed. But it's also a biennial crop, meaning it doesn't flower the first year, it flowers the second year. So you're talking about long time frames in order to do seed production. I visited with a seed company that was planning its onion production schedule, and their spreadsheet went out at least 10 years, because you have to think of the timeline of getting the parent lines. You have to grow the parent lines, and you have to produce seed of that somewhere. Then you collect those, and then you have to grow those in a location where you make the hybrids, and so on, and then those have to be prepared and go on to be sold and so on. And so if you have a 2-year cycle in the field every time, you can imagine that takes a long time frame to accomplish all that. Multiple locations around the world, all kinds of logistics involved in all that. So it's quite I think something that I tend to tell people. People to say, well, why shouldn't farmers just save their own seed? Why do they have to buy seed from these companies and so on? And my response to that is, why don't you just build your iPhone on your kitchen table? I mean, could you do that? It's a very complicated thing to do, and seed production, particularly these days, is equally complicated. It takes years and years and lots of technical input in order to accomplish that. Well, the seed industry is a major industry in California agriculture, although less well known and partially hidden, I guess you'd say. Of course, seed production in general is on the order of maybe 3%, 5% of the area of the crop itself. And in vegetables, probably even less, because again, a lot of it is done in greenhouses in very specific locations. So as a visible industry, it may not appear to be so big, but it's very critical. And California is one of the major locations around the world for vegetable seed breeding and production. 2 reasons. We have an incredible climate in California, or I should say climates, because we have so many distinct climates. We have the coastal regions, we have the inland regions, we have the desert region. We have no rain virtually in the summer, which allows us to mature and harvest very high quality crops with much less disease pressure and so on. The other thing, of course, is that California produces about half of the vegetables for the United States. And so we are a major production location. Breeders want to be producing seeds and varieties that grow well for our growers here. So that combination of ideal growing conditions for producing seed themselves and a huge market for seed has resulted in essentially all major vegetable seed companies in the world operating in California. And we have cohort or an industry of very knowledgeable farmers. This is, again, growing seed is another level of complexity and expertise beyond growing a commodity. And it takes hand labor, it takes management, it takes additional roguing. Many of the seeds are certified, less so in vegetables. It really applies more to other agronomic crops such as sunflowers. We're a major hybrid sunflower seed production location, and all of that is certified. So there's a lot more oversight, I guess you'd say, and a lot more technical complexity to growing seeds. So we have the combination of climate, we have the investment of the companies here, and we have growers who have created expertise that is valuable, and equipment. For example, vine seed production, one of the few places in the world where we have mechanized harvesting of melons and so on, by machine, because of the scale that we have here for it. So all of those things have created a very big industry. The California seed industry as a whole sells primary productivity somewhere around probably $800 million a year. There's all kinds of opportunities in the seed industry. And it's not just breeders and farmers. In between, for example, a lot of plant pathologists are involved in the seed industry because, again, plant pathology is a very important part. There's laboratory people to do seed testing. The seeds are tested multiple times, so there's laboratory jobs doing that. There's laboratory jobs doing testing. There's phenotyping, huge roles now. So all of the, say, tomatoes or other crops that have that go into processing and so on, all of those processing characteristics have to be tested. That is the solids or the sugars that are in those fruits and so on at every stage of development. So again, all the way through the breeding. So there's a lot of that breeding effort. On the seed production side, there's enormous investment in quality control. You know, companies have huge laboratory setups to extract seeds. For now, a lot of it is DNA based, that is checking, is that really a hybrid? Is it really not? Is that the variety that I want? Does it have any disease in it? And so on. The amount of quality control and testing in that industry is remarkable. But also for the shipment of seed around the world now, there are phytosanitary rules so that you have to have tested and documented that you've, have either zero or some below some threshold amount of disease present, or you can't move seed. You can't move it around the world. So there's all of this background. Then again, beyond that, marketing. There's people out marketing. There's field representatives to go work with the seed growers. There's people doing what they do in every business, accountants, there's everything. We actually offer through our Seed Biotechnology Center a course called Seed Business 101, where we encourage people new to the seed business to come and participate in this class, work with groups, and this is across all job categories in the seed industry. Particularly the vegetable seed industry is one of the most complicated businesses there is. I've had CEOs who've come from other major businesses to work in the seed industry, so this is the most complicated thing they've ever seen because of the logistics, the international aspect, the complexity and the technology that goes into it. But then also it's agriculture. You have the variability of weather, and all the things that you really can't control, and that's what really makes it more challenging. Is it's not like a production line you can just turn it off. It's actually, as I said, it's years in advance that you have to be scheduling these things and projecting markets, projecting demand. You can imagine growing a whole warehouse full of seed, and then your variety loses out to another variety, and all that seed becomes really not very valuable. So managing that inventory is really a critical thing as well. What am I particularly proud of in my career? I guess that would fall into two buckets. If I could speak, to scientific area, I'm proud that we've been able, in terms of seed quality, to be able to be working in a time period where it's been impossible to go from DNA to traits. In other words, I was born the same week that the Watson-Crick paper came out. If you can believe that. That was the structure of DNA was discovered identical with my lifetime. And I wanted to be a molecular biologist from the time I was in high school. But to be able then to see that those technologies develop so that a person who's really more of a physiologist or a plant grower could use those tools. And so we've been able to do that. We've been able to identify all the way down to the specific gene, several traits that are related to seed quality. So on the science side, that's been exciting and very interesting, and then those can be incorporated in the crops. I think on the another thing that I'm proud of is to have been able to be a professor at UC Davis. That's I'm hugely honored by that opportunity, and it's been a wonderful place to work. But the fact that we were able to create the Seed Biotechnology Center 18 years ago, and then about 11 years ago the Seed Central organization, which expanded that to connect the university with the seed industry. And I guess one of the things I'm most proud of is the success of that. That is, we've been able to engage the entire seed industry. We've been able to demonstrate, I think, the value of academic research, and in particular connect that industry up with our graduates, not only at UC Davis but throughout the state. The Seed Central organization works with all the undergraduate universities in the state and provide opportunities for students to prepare themselves to be good employees, to meet with industry folks before they graduate, and then have jobs hopefully waiting for them when they show up, which creates more industry in our state, more jobs, more economic impact. So I can say those are sort of the two things that I'm most proud of.
Maintaining genetic diversity is a very important part of the seed and variety improvement process. Both university and federal government germplasm repositories and preservation programs for vegetable seeds are very active in California. These facilities not only maintain quality, viable seed resources for a wide range of genetic traits, but they also provide material worldwide to both research and commercial variety improvement programs.
Well, I'm Roger Chatelet. I'm the director of the Tomato Genetics Resource Center at UC Davis, and we're in our greenhouses located just off campus. So the Tomato Genetics Resource Center, or TGRC as it's known, is a seed repository, or sometimes called a gene bank, of different tomato varieties, wild species, mutants, and other types of stocks of tomato. And these are maintained in our greenhouses. You see a few plants that are growing here now. These are ones that we're growing this year just because we need more seed. We need to refresh the seed. And I'll show you how we do some of that, how we multiply the seed. And the other thing we do is we store the seed long-term in low temperature and very dry conditions to maximize the longevity of the seed. And we distribute the seed around the world to researchers and plant breeders who are interested in improving tomato for different traits. One of the things that plant breeding, sort of a fundamental idea of plant breeding is you're selecting out of a pool of germplasm or different varieties or breeding lines. You're selecting for improved lines that have traits of interest, whether it's higher yield or better fruit soluble solids or flavor, etc. So that requires a source of genetic variation. And sometimes breeders find that in the lines that they work with, in their selected breeding lines, there's not enough genetic variation. So for example, they might need resistance to a new disease. So in California, for example, there are new diseases that are on the horizon, and current varieties may not be sufficiently resistant to those diseases. And when they need new germplasm, they come to our center because we have the world's largest collection of wild species, of wild relatives of cultivated tomato, and that's where the new traits come from. And so we maintain this collection and as a resource for breeders for improving varieties towards the future. Our seed collection includes over 4,000 different lines, and this is roughly evenly split between wild relatives of tomato, mutants, so these are single gene mutations that have either been induced or just observed as spontaneous mutants, and other types of genetic stocks. These would be breeding lines that incorporate disease resistance in early generation tomato types, for example. So total over about 4,000 lines. Now for the wild species, each line is potentially quite heterogeneous. So they're mostly outcrossing species, so they require bees to cross-pollinate them, and that means that each of those lines are genetically heterogeneous, kind of like a human population. If you take two people, they're genetically different, and it's the same with these wild tomatoes. So when we say we have 4,000 lines, it's it's much more than that in a sense, because each individual plant of those wild populations is often different from all of the other individuals in that same population. So there's a lot of genetic variation in the collection. Of just 4,000 seems like a small number, but it's quite a bit of genetic variability. What you see here in the greenhouse is a selection of different lines that needed to be grown this year because we didn't have adequate seed, or the seed germination rate was too low. So we grow them out and then we pollinate them to produce seed. And we do the pollinations in different ways depending on what type of material it is. And so we have a large number of plants here. There are actually about 75 different plants from the same individual collection collected at one site in northern Chile.
To keep the wild germplasm supplies viable, Roger and his team do considerable, careful, and time-consuming pollination work by hand.
So we've been so far talking about tomato and the tomato germplasm collection at Davis. But for all of the vegetable crops and all of the major crops generally, there are similar collections around the world, in the U.S. included. The Davis collection is specialized just on tomatoes. There's another collection at Davis on clonal crops. So these things would be things like grapes, walnuts, all the prunus species like apricots, plums, etc. And there are several repositories like this around the U.S. that are part of an integrated national system called the National Plant Germplasm System. And so for all of the major crops that we rely on, breeders are using traits that are drawn out of the collections of the National Plant Germplasm System ultimately, and that's where the new traits are coming from, and they're bred into new varieties that we enjoy and can take advantage of. Just using tomato as an example, if it were not for collections such as these, we couldn't grow tomato in many areas where they're actually highly productive. Plant breeding basically involves two fundamental processes. One is making crosses between varieties, different lines, different selections, and then the other is growing things out and evaluating them and making selections. Make selections and you cross the best of those lines and you repeat the process. So it's a cycle.
Plant breeding and the crossing process involves two steps. First, the male parts of the flowers are removed in a process called emasculation. Then that flower is pollinated by pollen from another source.
I'm Jerry Sarimian. I'm a technician for the USDA ARS in Parlier, California. Basically, we collect seeds from arid plants for distribution to universities and breeders for research purposes. These are wild races that have been collected all over the United States, and when you're developing a variety, for a commercialized variety, you're kind of looking for the best of both worlds. So collecting these wild races and looking at the different disease resistance or yield or a number of other characteristics, how they adapt to different climatic conditions. These are all things that you take into consideration in breeding when you're trying to develop a commercialized variety. California is a long day, has a long summer, so it's a long growing cycle and compared to the other repositories across the United States. So basically they selected California to grow out all these seeds for the other repositories. We're growing it out, letting it go to seed. Like right now it's bolting. So in a commercial setting you wouldn't want it to bolt, you would harvest before that, but we're letting it go to seed. So you see it bolting, it's flowering, and so basically after that we'll collect the seed. I farmed for 24 years. I've been working with USDA here for over 18 years, and basically I like it because you get to experience a variety of growing different crops, and so you see things in wild races that you wouldn't normally see in commercialized variety.
In addition to the use of traditional breeding techniques and reliance on wild germplasm sources of genetic diversity to improve seed and crop cultivars, a number of new gene editing tools are now available that may allow us to more quickly and easily create new varieties.
My name is Diane Beckles, and we are in the Department of Plant Sciences at UC Davis. We have a number of projects, some that start with basic biology, or we want to be able to understand the role of genes in determining phenotype. And so for those projects, even though we're not going to have a cultivar at the end of 3 or 5 years, what we're doing is building basic information that we think will be useful for other people to do just that. The other thing that we're really hoping to do as well is to use the knowledge of genes to help develop biomarkers. So this could be a useful way of tracking certain produce. I should mention that we work on fruits and vegetables. These are perishable, and it's really important not only the traits that you breed at harvest, but how those traits translate from the field to the consumer. So how genes behave in that space from the field to your table is also really important, and we'd like to understand the role of genes in determining quality and shelf life. One of the traits that we're really fascinated by is called post-harvest chilling injury. And you may not have heard about it before, but it's actually really important, and it helps to explain why some fruit and veg don't taste as good when you buy it from the supermarket versus when you grow it at home. So if your fruit, vegetable, or even tuber originated from a tropical or semi-tropical region, chances are if you put it in the fridge, it's going to be damaged. It may not taste as good, it may not look as good, and in some instances, the spoilage may be accelerated, may be sped up. That's contradictory. You put things in the fridge to enhance shelf life, but for tomatoes and basil and nectarines and peaches, if you put them in the fridge, then you're actually harming potentially these crops. So we have some candidate genes that we think could be important in explaining why some of these species are sensitive to refrigeration. And we're starting the process of looking at the function of those genes in wild species and in the species that you would get in the supermarket, and transferring genes between these different individuals. And in so doing, we really hope to get a better understanding of this chilling injury problem. So this could mean that you can put your tomatoes in the fridge and not worry about having them taste mealy or not having a good flavor. And for producers and farmers, they would actually be able to use a fridge refrigeration for a better shelf life. Traditionally, when people have done basic research on produce, it was hard to imagine that you could take that gene and edit it or manipulate it and have it be sold. But now with with the advent of gene editing techniques, it is entirely feasible that you can study a gene, determine that it has an important role to play in the crop, and in a way that's fairly cheap, fairly easy, and then without all the cumbersome government regulation, get it from the lab to your table. You could take this knowledge and use it in a traditional breeding approach, but we're not limited to that now. And this is important. Traditional breeding has taken us from, crops that were low yielding and didn't have very exciting attributes, 50, 75 years ago, to this amazing array of products. But with climate change, with sophistication in terms of what consumers expect, it's good that we have additional tools to take information about genes and to be able to modify them.
The process starts with what we call transformation. So it's basically we introduce the gene of a foreign gene from another species into the tomato plants by the help of a bacterium called Agrobacterium tumefaciens. And then we have to regenerate that initial tissue into a whole plant. I have to wait until the plant grows, it makes fruit, and then I harvest the fruit and collect the seeds so I can grow those plants again, extract the DNA, and check if those plants are transgenic or not. Yeah. Here we are not trying to produce or generate a tomato that is going to be available for the farmers. This is a proof of concept and it's basic science.
My name is Rick Falconer. I'm Managing Director at Reichswan Seed Company, Reichswan USA. And we're here in Salinas Valley, close to Gonzales, California. And we're at our research farm. At this facility, we breed and select leafy vegetables primarily. So lettuce, spinach, brassicas like broccoli, cauliflower, and cabbage. Our company does a lot of different vegetables, asparagus to zucchini, and we work very hard to develop new varieties. Variety development is our biggest task, and through plant breeding, it is what our everyday job is. And it's basically, to really bring it down to the simplest form, it's selection. It's evaluating different varieties, either thumbs up and thumbs down, and we're doing that constantly on a daily basis, thousands of times during the day, and evaluating different traits of varieties. Some of it is yield, pest resistance, insect resistance, but also we do a lot more these days in consumer traits. So flavor is really big now. Appearance, size, unique shapes and colors. And so, the industry is very dynamic. We have a lot of scientists working with us, but we have a lot of business and development people as well. Yeah, this is a typical research farm that Reichswan has and every seed company out there. Out here we have zucchini, we have beets, and several varieties of lettuce. And this is what we call a trial. We use that word a lot, a trial. And there may be 12 varieties or there could be 250 varieties. And in this process, we're measuring and comparing to standard varieties and going through and taking the various data. On zucchini, we're taking yield, so we're harvesting that, weighing it, making evaluations on things like color and skin thickness. On lettuce, we're cutting them, we're weighing them, we're looking at the different diseases and pests that are hitting those varieties and marking all that data down. So data collection is really our main deal, is being able to put that into the computer and crunch the numbers and being able to, again, select the best varieties for whatever traits we're looking for. For spinach, again, as simple as it is, it seems, there's processing spinach, there's bunching spinach, there's baby leaf spinach, and there's a lot of diversity even within a crop like spinach. This is a Savoy variety, but it's been hit with downy mildew. So you can see it gets necrotic on the underside. They also call it blue mold because the mildew sporulates, and those are millions of spores on the back of that leaf, and those just get disseminated by the wind and it just spreads like wildfire. So this is our seed warehouse, and this is all packaged spinach seed. And after we grow the crop, we package, we size the seed, we treat the seed, or we don't treat the seed if it's organic. These are all pallets of spinach seed. Each one of those bags on the pallet has 1 million seeds in it. That's how we sell spinach seed, by the million. One bag is worth about $300, give or take, depending on the variety. And each pallet generally has about 60 bags per pallet. So one pallet would be around $18,000. So you can see the seed is expensive. A typical grower will plant on an acre about 3.5 to 4 million seeds to the acre. So you can see it's well over $1,000 per acre for a grower to grow spinach seed. A typical seed label contains the variety name. This one's name is Platypus. Before it was commercially on sale, it had an experimental number, and that's the number that Reichswan gives it, 51707. And in the early phases of development, it didn't have the commercial name yet, but it was known through the selection process, through all the phases, as 51707. We have here a designation called precision seed. It's basically sized seed. So it goes through a series of screens and sizing equipment. This particular one shows that the range of the seed is 2.75 to 3.50 millimeters. On a seed lot, you're going to have the genus species of the crop. You're going to have the germination. We're required to list the percent germination. This bag is 91%. It's also going to list the test date because it's important to know how old the seed is. The germination test on this particular lot was on December 14th, 2017. It also gives the seed count, so the number of seeds per pound. And then other various pieces of information that we're required to put. This is untreated seed, so there are no fungicides applied to this seed. So this seed is not organic seed, but it can be used with organic growers because it's untreated. Spinach seed grows in many different areas of the world, but 60 to 70% of spinach seed is grown in Denmark. Denmark has, number one, the infrastructure and the expertise of the growers in that area grow good spinach seed, but also it's at a high latitude. And that's required on some varieties because
with those long days, the spinach plants bolt and they'll go to seed. If you plant some of those varieties in lower latitudes, let's say here in California, it'll be a long time before it sets seed or flowers. So not only Reich Swan, but most other seed companies have some presence in Denmark to produce spinach seed. We also produce in the US, in Washington, Oregon, on some varieties. Those are faster bolting varieties. So we do produce some in the States.
Tony Turkovitch of Winters, California, is a vegetable seed producer who grows a variety of vegetable vine crop seed. His operation is highly specialized and relies on a range of growing, harvesting, and seed processing techniques.
I'm Tony Turkovitch, and I farm here. Button and Turkovitch is the farming operation in Yolo County near Winters, and we're in a cucumber seed field, a hybrid seed production. When we planted this, we planted a male row, which is the row I'm standing on that we've destroyed already. And then once it came up, then we planted 4 rows of the female seed that the buyer wanted. And so we had to separate them so that they would bloom at the same time, because we want the pollen from the male flowers in the male row to pollinate the female plants. So once it was done pollinating, then we came in and we destroyed the male row, because that would not have the hybrid seed that we want. Some people do a 3-bed. We're doing a 4-bed and 1. Some do 3 and 1. So once it matures, then we harvest. We do this for cucumbers, squash, hybrid watermelon, any of the hybrid melons or things like that. We also grow open pollinated, but that would be just one seed on the whole thing. It wouldn't have the male female, just one set. And then the same concept though is done in the onions and the sunflower that we grow. The sunflower females are male sterile, so they only can take the male flowers. On some of the male pollen, on some of the vine seed, we have to use a growth regulator on the females so that the male flowers are not potent, and so for a period of time we'll spray for a period of time so that during a 2-week period, whatever pollinates we know can only have been pollinated by the male pollen on the male row. Depending on which seed. Sunflower, there's more demand, so a lot of people grow the sunflower seed. The vine seed, the amount of seed that is needed is not that great, so the acreage that's available is very limited. So it's been over a period of time that we've built this up with different companies that need the seed, but it's not something that every farmer in California could put in their rotation, because there's just not that much demand for the seed. So we bring the bins of washed seed in from the field, and we unload them, usually 2 bins into the dryer, and through the top door when it's in line, and then the seed is tumbled slowly. We've got in front a big fan and a burner that puts heat in if it's required. We maintain around 100 to 110 degrees, and then the hot air blows underneath. It's in the plenum and then up through the dryer, and the dryer, that's a screen. The whole drum is a screen, so the air flows up into the dryer through the seed, and then over a period of time it's dried. So it takes, depending on the seed and the seed size, from 8 to 20 hours to dry.
California farmers also produce lettuce seed generally in large fields where blocks of different types of seed are first grown and then machine harvested using highly specialized equipment. A great deal of work is then required to clean the seed and to separate it from other parts of the plant using rakes and blowers that blow away the non-seed material in a series of cleaning operations right out in the field. California's seed production and variety improvement industries and the fundamental research program that underlie these industries are of immense importance to the state's sustained preeminence in vegetable production. There's a very wide range of aspects to the public and private sector contributions that many highly skilled, knowledgeable, and dedicated contributors make to vegetable seed and variety improvement efforts every day. From plant breeders and geneticists who identify basic variety improvement mechanisms and biological pathways, to commercial seed companies that further refine the selection and improvement process, countless trials, and their marketing of seed worldwide, to the farmers who grow the seed to increase supplies. There are many critically important opportunities for new contributors to become part of these dynamic and exciting efforts.